All right. Anybody start baking for Thanksgiving yet? No. Pie is gone yet? Yes, we'll be hosting Thanksgiving. Plenty of family coming over. What? Yeah.
The dishes seem worse.
Yeah, the dishes aren't great, but it's all right. We don't have to travel anywhere. So we'll probably eat some proteins. So normally we think of protein as this one thing, like a part of your food. You look at the label, it says protein, but it's not really one thing. We should really talk about proteins, because what makes them be in the same category actually has very little to do with their function in the body. Proteins do all kinds of different things in the body, and much like lipids and carbohydrates, it's not what they do that makes them similar, it's what they are that makes them similar, their structures. So we're going to look down at the molecular level and look at structures of proteins. So what are proteins made out of? Somebody knows what proteins are made out of. What? Amino acids, yeah. So proteins are made from amino acids. So I guess we should talk about what amino acids are, because then that's going to make it a lot more sense when we see what proteins are. All right. So amino acids are different molecules that have very similar structure and only really differ in one point. So an amino acid we're going to put a carbon in the middle. We're going to put an H off to the right. And an NH3 off to the left. And because it's an NH3 with another bond on it, that actually makes an ammonium and makes it positively charged. And we'll talk about why that is in a minute. Then off the top, we're going to have a carboxylic acid, but like in the soap, rather than being an acid, this is actually going to be a carboxylate. So the conjugate base of the carboxylic acid. And then the last position down here, we're going to call R. Remember, R kind of means like wild card, like could be whatever. And this is the structure of all those 20 amino acids. They all have that same part structure except for the R. They differ on what's coming off down there. Everything else is the same. Okay, so one thing to note about this structure is this is actually a Fisher projection. So the fact that that nitrogen is on the left ends up being important. And that tells us that these are L amino acids. When we build those, we have to build it that specific way where the middle two things are kind of coming out at you, because that is indeed a Fisher projection. Now that said, we're really only going to deal with the L amino acids. So just like the sugars, we said there are D sugars and L sugars, but really only the D sugars participate in metabolism and can do that kind of stuff. Similarly with the amino acids, it's actually the opposite though. All of our proteins and all our structures that use amino acids are all built from L amino acids. So we're not really even going to look at the D amino acids. You can make them, but they really don't. They can't really be. Well, it's not 100% true, but for the most part, you can't get the D amino acids from nature. So we really focus on the L amino acids. So depending on that R group, we have this big list of different ones, and you don't have to memorize them or anything. But I do want to show you what they look like. So let me see if I can get this picture here. Just pull this over here. So there's all our amino acids, and they're grouped into a few different sections that you could group also. So most importantly are nonpolar versus polar. As we've talked about a lot with the last few chapters, whether things are nonpolar or polar has a big influence on what exactly those things are going to do in the body, where they can go, where they can't go, can they get in the cells, can they whatever. So that's the same with amino acids. So we want to be able to tell what are the nonpolar amino acids and what are the polar ones. And when we talk about that, we're really only talking about these side chains or R groups, because the rest of the structure is always the same. So if the side chain or R group has all this nonpolar stuff like carbon and hydrogen, then we're going to call those the nonpolar group, the nonpolar ones. So glycine, alanine, valine, leucine, methionine, and isoleucine. Again, you don't have to memorize all those names. If you go on and take some biochemistry, or if you're going into a health program and you have to take some types of stuff like that, physiology, you may have to memorize all these at some point, but you don't have to for this class. And then you've got the polar ones. So keep looking over on the left here. See how these are more polar? These have things like OHs, NHs, SHs, hydrogen bonding capabilities, that sort of thing. So this is actually a little bit wrong. It's actually this proline is, because it's kind of wrapped around here, it makes it polar. But anyway, those are the polar R groups. And then there's also a bunch of them that have charges on their R groups. So those are going to be really even more polar. Positively charged R groups, negatively charged R groups. And then they actually separate, some tables do it this way, some don't. But these separate out these aromatic R groups as well, because those are going to behave a little bit differently too. So those are non-polar, but also have this aromatic component to them. All right, so that's basic structure of amino acids. Now let's look at why we drew them with these charges, because depending on where you look, you may sometimes see amino acids drawn like this, with a neutral amine and then a carboxylic acid. So that would have been a lot nicer to draw it without the charges, and just that kind of just looks like a regular organic structure. But that's not actually how it would look in any solution. The reason being that carboxylic acids are acidic, so they can give up their hydrogen. We've talked about that a lot. In the same way, ammonia or nitrogens are basic, and they accept that hydrogen. So if you have the same molecule that has an acidic spot with the hydrogen and a basic spot that wants to take the hydrogen, it's going to end up doing that. So it's sort of like the acid-base reaction, and we end up with the conjugate acid and the conjugate base. So rather than showing the structure on the right, we show the structure on the left, which is the conjugate acid of the amine, ammonium, and the conjugate base of the carboxylic acid, which is the carboxylate. So that the acid loses its hydrogen, and the nitrogen gains the hydrogen. And that's the typical neutral structure at a neutral pH. If we change the pH that this is existing in, that would change. So if we are in acidic conditions, we would put that hydrogen back on. If we're in basic conditions, we would take the extra hydrogen off. But in neutral conditions, we want a neutral molecule that has a plus and a minus in the same molecule, and so that's what we got. Let's save this. Let me save this for a second. All right. So there's our amino acids. Another thing you should know about amino acids, again, you don't have to memorize them, but just to be informed about it, because it will come up here and there, is that, I'm just trying to find the book again. One thing we're going to be concerned with in a minute is how these amino acids connect together. And because there's a bunch of them and they all kind of, a lot of them have these kind of big names, we often abbreviate them. So let's take a look at what the abbreviations for the amino acids look like, just so that we can know what we're talking about when we start talking about that. No, maybe not. Okay, here it is. So this is on page 638 in the book. You can pretty much figure these out by what they look like, at least the 3-letter ones. So most amino acids are usually listed by their 3 or 1-letter abbreviations. So 3-letter abbreviations, you can see there, they mostly look like the names themselves. Again, you don't have to memorize these abbreviations, but I may give you a table like this or give you some of the pictures of the amino acids and ask you to do some stuff with them, like hooking them together, making peptides, whatever, which we're going to do in a minute. When you're actually trying to analyze these things in the real world, you're often talking about proteins that are made up of thousands of amino acids. And if you're talking about thousands of these things connect, hundreds or thousands of these connected together, then even a 3-letter abbreviation becomes tedious. So that's when we use the 1-letter abbreviation. So if you hear about things like protein sequencing, that means knowing what order the amino acids are connected in. And so you would do all this experimentation, use some special types of equipment, and get this big readout that looks like, G, C, D, T, T, G, T, A, H, Q, N, R, whatever. And that's the list of the amino acids in the particular order. And then you can search for matching groups of letters if you're interested in how a particular protein is like another one or something like that. So that's what these abbreviations are used. Okay, so let's look at hooking these things together now. So to get from the amino acid level to the protein level, we have to connect them. And they connect by something called a peptide bond. And we should note, this is also known as an amide. That's a functional group we've already studied. It's the exact same thing, but when it's amino acids and proteins, it's called peptide. All right, so let's put some amino acids together and look at how they connect. I'm going to draw this all out like old-fashioned Lewis structure style, where we see all the different atoms, so we can really carefully track what's going on here. I'm also not going to worry about stereochemistry or orientation or angles or whatever. I'm just going to list them kind of like when we first started learning about organic structures. All right, so there's an amino acid. This one happens to be glycine. Glycine is the smallest amino acid because it's the one where the R group is just hydrogen. It is also the only amino acid that doesn't have an L or a D, because remember when we were working with those models, if you have 2 parts that are the same on a carbon, then they're superimposable no matter how you switch things around. So L and D don't sorry, I was thinking I said R, but I meant L and D don't tend to matter for this one. Okay, so there's my glycine, and then next to it, I'm going to make a molecule of alanine, same way. So alanine is different from glycine in that its R group is CH3 instead of just an H. And everything else is the same. So notice those 2 structures only differ by that R group coming off of that carbon. So alanine is abbreviated ALA or A, and glycine is abbreviated GLY or G. So we're going to connect these to make a peptide. And to do that, we're going to do the same type of reaction that we did when we made the glycerides. We're going to join to make water, pull water out, dehydrate, and then connect what's left over. So the H2O that we're taking for water is going to be right here. 2 H's from the N and then the O from the carboxylate. And then we have to connect what's left. That. Okay, let's redraw that. Everything else stays the same. Remember, when we do these types of reactions, just like with the glycerides and the glycosides and everything else, you don't mess with any of the rest of the molecule. You're just changing where that connection happens, and everything else is staying the same. So now, instead of the oxygen connected here, we're going to connect to the nitrogen of the alanine. That nitrogen still has one of its hydrogens, because 2 of them were taken to make water. And then it's going to have the rest of its molecule here, the CH3, and the carboxylate. And the byproduct here is water. Okay, so we made a bigger molecule, but notice the ends of that molecule. The left end has an amine or an ammonium, just like an amino acid, and the right has the carboxylate or the carboxylic acid, just like an amino acid. So this thing really can then behave like a giant amino acid and keep connecting to more of them. And that's how amino acids can build up proteins. So for this one, we would call this one a glycine-alanine peptide. So we would kind of connect them with a hyphen like that to show that that's indeed what we're doing. We could also, if we were sequencing something big, it would go by just letter abbreviations, which would be GA. Doesn't mean a whole lot there, but if you can imagine, if you had like a string of 1,000 of those, that would be all those different ones hooked together. And you can see the peptide bond that we made right here. And that is the same as the amides that we talked about a couple weeks ago when we talked about functional groups. Okay. Does everybody have a computer or some sort of device around them still? Something they can look at, or you can look at somebody's next to you. All right. I want you to, in a minute, pull up a list of the amino acids. You can do it directly from the notes here. They're already updated from Blackboard, so you can look at this specific list, or you can just pull up any list of the amino acids. Okay, and I want you to practice writing a couple peptides, so combinations of amino acids. So try this tripeptide of alanine, glycine, and valine. And then try another tripeptide of tyrosine, serine, and histidine. So you got to find the structures of those amino acids, and then hook them together to make the tripeptides. that track. But yeah, there are some good.
Yeah.
Yeah. Yeah. By those three. You may need to look up the abbreviations too.
Do you need abbreviations?
But I mean, all of the ones I put here are just the first three letters of whatever amino acid. So as long as you have names, you can probably figure it out. Bailey. Yeah. This is the first one. I didn't know that too. Because here. Yep, that's enough. again, once you throw in some peptides, share with people around you and see if your peptides look similar, if they look connected the same way or not. Careful there. Yeah. Looks like some good peptides out there. I'm going to start drawing the first one, but don't look at it. Just working on what you're working on for now, and then you can take a look and check it. So it should look something like that. You see the peptide connections there, and the R groups. There's a little thing in the book where once you practice this, you'll notice there's kind of a pattern. It always put the end terminus on the left, where it stops at the end on the left. Then it kind of goes CCN, 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 CCN until the very right end, which then gets the carboxylate. And then you have the R groups coming off that first C. So every time you do the CCN, that first C always gets whatever R group it is. So you can relatively quickly draw these out that way. And then just put in the various R groups that you need from looking them up on the table, or if you know them, you can just write them. All right, so I'll leave the second one for you to do, and let me know sometime if you want to take a look at it. All right, so the next piece of this is what happens when we get quite a few more, and what does that actually start to look like in 3 dimensions? We talked about how proteins are these big molecules, so we're slowly building up from the smallest pieces of them to then what they look like as we zoom out and zoom out and zoom out. So let's take a look at this picture from the book, just because it starts to become kind of not useful to draw it once it gets big enough. So here is a 1, 2, 3, 4, 5, 6, 7, 8 residue peptide. That word residue is often used to mean each individual amino acid. So if you've got, what did I say, 8 of them, 8 residues, that would be whatever you call an octapeptide. So we've got several of them hooked together. And here, with this modeling, the way they've done it here, you could at least see the zigzag chain. Even though we've been drawing it straight across, it does zigzag as you'd expect. And because of the 2 carbons, so, well, I guess it's really 3 zigs in the chain, right, in between. That's not making any sense. Let me just count it out and show you with the mouse. So here's a residue coming off, or here's an R group coming off of this one. There's our phenyl group from phenylalanine. And then we go 1, 2, 3, and there's glycine. The R group's an H, so we don't see much there. 1, 2, 3, there's alanine, so there's the CH3 coming off. And then 1, 2, 3, right? There's our leucine, so there's that isobutyl group coming off of the leucine. Anyway, the point of that is that the residues end up kind of coming off opposite sides. So they go below the chain, above the chain, below the chain, above the chain. And even though there is rotation around some of the bonds in the structure, though not all of them. So this doesn't end up being a straight chain. And if we built the models, we could see this. It actually has a couple different choices of what to do, and that's called the secondary structure. So let's define first the primary structure, and then we'll take a look. We'll look at a couple of videos of how that works. The structure of proteins, because they're so big, is often broken up into 4 parts: the primary, secondary, tertiary, and quaternary structures of the proteins. The primary structure is what we've just been talking about. It's the sequence of amino acids. So which amino acid is connected to which one, and what order are they connected in? As you can probably figure out from what we just did, the order really matters. If you have gly-ala, that's a different peptide from ala-gly. They're separate. It matters which is on which side. So the primary structure tells us which ones are connected to which and in which order. Okay, so we start to zoom out, and we get to the secondary structure. And the secondary structure tells us how these long chains start to kind of fold on themselves and make new shapes in larger repeating structures. So the secondary structure are the large repeating structure and I guess it's best to just kind of show you what these things look like. So there's a couple main types that we're going to look at. The first is called the alpha helix. And the second is the beta sheet. And rather than try to draw these for you, I'm going to show you pictures of them being drawn a little bit nicer, and then we'll look at a video of it as well. What? Okay, so let's take a look at this picture that the book shows here. Okay, I get to it. Okay. So let's see. Let's put it in the notes so we can mess with it more. All right, take a look over on the left here. If you look closely in there, this picture's on 643 of the book if you want to follow along also. But if you look in there, you see a chain of amino acids, and all the R groups here are removed on this particular one. There's no R groups shown here. So you just have the amine, or the carboxylate, the carbon, then the amine connected to the other carboxylate. So there's the other amide or peptide, carbon, peptide, carbon, peptide, carbon, peptide. And the reason they wrap around in this particular way is because when they're in that orientation, the hydrogens from the amine can hydrogen bond to the oxygen from the carboxylate on a couple residues down. So that's shown in the next picture here. So I'm going to try to zoom in on this. So here's a hydrogen from the N part of the amide. And that hydrogen is hydrogen bonding with an oxygen from the peptide, or from the peptide, from the carbonyl, that is what, 1, 2, 3, 4 residues away. I guess 3 residues away. Then the same thing's happening on the next one. The next one's interacting down here. And then this one's interacting over here. And this one's interacting below. And those strong intermolecular forces, those hydrogen bonds, hold this in that particular orientation. Because if that were to untwist, those things would no longer be lined up and would no longer be able to attract each other. So when it twists in this particular way, when the bonds rotate just right, all those kind of lock together, and this ends up being a pretty stable structure overall. The aggregate of all those hydrogen bonding interactions makes for a pretty strong structure. If you then look kind of down the middle of the tube, which is what's in this picture over on the right, you see the main part of the chain, the NCC, NCC, NCC part, right here. And then all the R groups, whatever they may be, are around the outside. They're kind of poking out or pointing outwards in that structure. So that means that depending on what those R groups are, that's really going to control what this whole helix looks like from the outside to other molecules. So if it's more polar, if it has a bunch of the polar or charged R groups, that's going to look more attractive to, say, water molecules to dissolve it. If it has more of the non-polar groups, well, then it's going to go through lipids, interact with lipids a little bit better. So all of those things are going to be controlled by how that structure holds itself together from that chain, and that's called an alpha helix. Any questions about that or how that works? Okay, then let's take a look at the next one, which is the beta sheet. We'll put this image over here too, so it's not too cluttered. All right, so in a beta sheet, you have these kind of zigzagging sheets, really, of hydrogen bonded chains. So let's see if we can zoom in and see those hydrogen bonds again. In this case, here's your CCN, CCN, CCN going on. There's your oxygen from the carbonyl. And in this case, it's pointed kind of straight across to another parallel chain. And the H from the nitrogen and the O from the carbonyl are again involved in this hydrogen bond, which holds these things together. So you've got these two chains like that, kind of lined up like that. And then the R groups in the beta sheet are going to be pointing up or down from the sheet. So you got this kind of sheet of these chains that are interacting like this, and then the R groups sticking up and sticking down. So again, the outside of it, what other molecules would see as the outside of it, are going to be controlled by the polarity of those R groups, what those R groups look like. In this particular example, these chains are made completely of glycine and alanine, and they're kind of color-coded with these squares versus diamonds. So you've got you can see the legend here. The glycine are the squares, and the alanine are the diamonds. And so you can see one kind of ends up, if they're alternating, one's always going to end up, and the other one's always going to end up down. And that's how these sheets can stack on top of each other, by the residues sort of fitting together in each other's spaces. When you see this drawn in depictions of protein structure, both of these, we don't usually draw them this way because we're not zoomed in this much. We're not just saying, we're not seeing the individual atoms. That's why we use these ideas of alpha helix and beta sheet, so that we can, again, zoom out and think about the larger structure of the protein and what that might look like. So I found a video of showing these things, and it may be clearer than what we saw before. So here are your amino acids. Again, there's your chain. And the side chains are the R groups. That's another word for the R groups, the things coming off. Okay. So you can see that kind of helical structure there with the R groups coming off the outside. And then I think they're going to show, yeah, they're going to show the hydrogen bonding here between the N, the hydrogen from the N and the carbonyl. That's what makes those connections and holds that helix together. Okay, so now we're going to add some more residues onto it. Again, you can see those side chains on the outside of the helix. And if we look straight down the middle of it, you see that main chain, again with the R groups or side chains out the outside, and the hydrogen bonds forming that backbone in those green dotted lines there. That makes for a pretty strong structure. So when we actually see this in a protein structure, as you'll see when you look at some larger structures, all you see is that ribbon, what they call the cartoon there, just that ribbon-looking thing. Usually the R groups aren't shown, the inner molecules aren't shown, but if we see that twisted ribbon, everybody knows immediately, okay, that's an alpha helix, this is actually what's going on. So that's why we talked about the secondary structure, so we can kind of have a shorthand of knowing what's going on and what it looks like. So if we just see that in a protein, we know what that is. Okay. Same idea with the beta sheet. You can see the flatter structure here now of the aligned chains. So zooming in on just a couple of the residues and taking away the R groups, you can see these different strands, and you can see how the nitrogen, hydrogens, and the oxygens are connected by these hydrogen bonds, which hold these sheets together then. So then they extend it further. And so beta sheets are drawn like this with these flat arrows. They can either be connected parallel or anti-parallel, opposite. And okay, so you can see the side chains. Let's stop there for a second. You can see the side chains with the R groups above or below the sheets. The other thing I wanted to note here, let's go back a little bit. So they're drawn as arrows. The head of the arrow, like the pointy part, is the carboxylate, and the tail is the amino part. So you're always going to have an amino side and the carboxylate side, and two of these sheets are going to be hooked together, and then multiple ones can be hooked together also in either orientation. I think that's it for that one. Okay, so that's our secondary structure: the helices, and the alpha helices, and the beta sheets. And those can come together now in the next level, which we call the tertiary structure, or how this protein looks when it's all made up of those other pieces. Let's define that. So the tertiary structure is going to be the three-dimensional shape of the entire peptide chain, which could be hundreds or thousands of amino acids. And this is going to have those alpha helixes inside of it, the beta sheets inside it, and other stuff too. And those are going to be connected to each other by a few common ways. So let's see, where's my pictures again? Here they are. So let's look at a big whole protein structure. So there's an entire, this is a depiction of an entire peptide chain. So you can see areas where it forms a helix. You can see areas where it's in the flatter sheet. And then those individual areas are connected together and shaped by the interactions between totally separate parts of the chain. So take a look. There's 4 different types of interactions that are important that we're going to talk about. Let's take a look down here first at one that we know well, and that's hydrogen bonding. So we talked about how hydrogen bonding holds the helices and the beta sheets together. But the R groups, if they have these types of groups on them, remember the R groups are sticking out from the helices and the chain and the sheets, those can connect via hydrogen bonding also. And so here you have these things that are maybe 100 residues apart from each other or more, but they're kind of folded onto each other and holding that protein together in that way. So hydrogen bonding is one way. They can also be held together by nonpolar dispersion forces or hydrophobic interactions. So aromatic rings can attract each other. You see that one below there. Also just regular old carbon type stuff. So the nonpolar amino acid R groups can attract each other. And those attractions help hold those in place as well. 2 other types of forces that hold these things together are what's called a salt bridge, which is really just ionic forces. So you have a negatively charged R group and a positively charged R group, and those 2 are going to attract each other just like positive and negative ions do. And then the last one actually looks like the stronger bond, but it's labile, it can break, and that's disulfide bridges. Any of the residues that have sulfur-containing R groups can be linked into these SS bonds. Those SS bonds aren't really much stronger than an intermolecular force, so they can be broken and kind of go back and forth, but they can preserve that structure as well. This is how, actually I don't know if it's still true, but it's how if you get your hair curled like a perm, everybody know what that is? Or like curls up. That's basically what you're doing, is you're using chemicals to form those disulfide bridges and pull those hair proteins together and like force them to curl up. The opposite is true of things like hair relaxers that break some of those interactions and get the hair to smooth out and straighten out. Wow, it gets really quiet when that fan turns off. Thing's loud. Yeah.
I'm trying to tell somebody to get out of here early.
Is that what it is?
Yeah.
Yeah. We're almost done. All right, so there's our overall whole protein. And every little bit of that influences what that protein does. So we didn't really talk about protein function that much, and that's because proteins do everything that you can think of, really. I mean, there are structural proteins that give your body structure, your cell structure. There are transport proteins. We talked a little bit about the lipoproteins that transport nonpolar things through the bloodstream. There's things like hemoglobin, which is a protein that transports oxygen. There's enzymes that do all the metabolism in your body. Those are all proteins as well. And any one of those things, if this whole thing isn't right, that's going to go wrong. We didn't really talk about the quaternary structure of proteins. The quaternary structure is when you have some proteins are actually made from multiple ones of these that then end up coming together. So if you have multiple chains packing together, that's called the quaternary structure. So that's kind of the fourth level. So anything that disrupts these kinds of interactions can really cause problems for those proteins that need to be doing what they need to be doing. And one of the ways of causing that problem is one that we did on purpose a bunch, which is called denaturation. Denaturation is interfering with any of the forces holding the protein structure together. Which then causes it, oops, causing, I should say. Causing it to unfold. So, thinking back to the last few labs, what are some things that denature proteins? What are some things you can do to proteins? Alcohol is one. So last week, you put the albumin protein with ethanol, and you watched it kind of turn white, right? Those of you who've got it to work. And then the greater concentrations of ethanol cause more and more of that coagulation. So what's happening there is the ethanol is changing the intermolecular forces because it's not water. So it's interfering with some of these interactions. And not just these four types, but also the hydrogen bonding of the helices and the sheets too. So it's actually pulling all that stuff apart and pulling the chains, the peptide chain just into one big, long, disorganized thing. That's the denaturation part. We don't really see that part. But then once it's all stretched out, it's such a big molecule that it starts to interfere with the other proteins around it. And then that's called coagulation, and that's when we start seeing that kind of white stuff that was forming in there. Those denatured and coagulated proteins start to fall out of solution. So ethanol was one way to do that. What's another way to do it? Heat was another way. So if you heat these things up, interactions are weaker than the covalent bonds. Once you get above a certain temperature, those interactions are going to break. And again, you're going to stretch that thing out and allow it to coagulate. What else? Think back to what? Maybe I'm not sure about that one. If you could physically breaking it up. Maybe. What else did we do though? I did that. Well, think about like the milk lab was the other one where we were doing stuff with proteins. How did we coagulate the proteins in the milk? Remember? No memories. We used acid. So you acidify the milk, and that causes it to get all lumpy, causes those curds to come out. And that's exactly the same process. The acid interferes with the interactions, the protein stretches out, and then coagulates, and that's where we got those kind of chunky, cheesy things. And we use that in things like cooking. Like if you're making yogurt, you want to ferment some milk to make yogurt. You have to do a couple things. You have to heat up the milk first because you have to denature the proteins so that they can then form that nice kind of gelled structure that we like in yogurt. And then the fermentation actually acidifies it, and that's what causes the eventual end flavor and everything. So let's see, what else? Did we miss anything? So we talked about pH. Yeah, and you're right. Mechanical stress, stirring, whipping, or shaking can disrupt those interactions as well. Yep, that's the name. So that's how you make it. That's why you, when you beat egg whites, they end up forming that like stiff structure because it's the proteins that stretch out and then coagulate. All right. So the last thing about proteins. Have you ever heard the term essential amino acids? What are essential amino acids? You say they're not necessary?
Not necessary, but they're like
Kind of.
wait.
Yeah, it's a little bit of a confusing term.
They don't make it.
Right, exactly. We don't make those amino acids. So essential amino acids are, we don't make them. Our liver makes amino acids. And so these are ones that we need to get from our food. And I think there's a list of them in the book. The essential ones are isoleucine, leucine, lysine, methionine, so the sulfur ones. Well, not all the sulfur ones. No, not the sulfur ones, sorry. Leucine. Phenylalanine, threonine, tryptophan, and valine. So most of a lot of the nonpolar ones. We don't make them. We need to get them from our food. So have you ever heard the term complete protein? A complete protein is a food source that we can get all those essential amino acids from. And it's relatively easy to get the amino acids you need, assuming you have access to enough food. So complete protein is food. So generally, again, unless instructed by a doctor or something, there's no need to supplement individual amino acids for any reason. If you eat complete protein, you're getting these big proteins, these huge chains, that your body then breaks up into its constituent amino acids and then uses to rebuild whatever. Complete protein would give you all of those that you need. Animal proteins are all complete proteins because we're also animals. The only complete protein from non-animal sources is soy. Soy is a complete protein. You get all the amino acids you need from soy. But other proteins or other sources of protein like beans or grains or things like that, if you only had access to that one family, you would not get the amino acids you need, and you could have some pretty severe problems from that. But luckily, if you have a mix of those, it's not an issue. So like beans aren't a complete protein, and corn isn't or rice isn't a complete protein. Rice is mostly starch anyway. Corn maybe. But if you mix those together and you eat a diet that has all those, then you've got all the protein you need. So so it's for most people who have access to enough nourishment and can eat a relatively balanced diet, there's no danger of not getting the amino acids that you need. But for a lot of people in the world, it is just because they don't have access to enough food or enough of a variety of food. All right, any questions about proteins?
I
Can you also get that stuff from like a powder protein by that GNC stores?
Sure. Yeah. So if you, well, I mean, they sell all kinds of powder that is, a lot of it is just nonsense. But
Like, is it real though?
but
Like,
protein is, I mean, protein is protein. Yeah. So it's usually whey protein, which comes from milk. So it's usually milk protein, which is basically like what we did in lab. You isolate the protein from the milk by coagulating it and dehydrating it or whatever, and then you powder it up, and then you can reconstitute it with water and drink it. But yeah, it's protein. Protein is protein. The other thing people don't sometimes get about protein, though, is our bodies metabolize it just like any of the other macronutrients. So we turn it into energy if we don't use it for specific protein-related stuff. So people who are athletes, who are building a lot of muscle, need extra protein to accomplish all that stuff. But it's become kind of a thing now that you just always want to eat more protein, and everybody always says, oh, like all the products advertise more and more protein. But if you're not doing those sorts of activities, then you might as well just be eating, whatever, anything else. It just doesn't make that much of a difference. You're just gonna turn it into calories anyway. Other questions about proteins? Yes. Let's take a look at it. So let's try to do this one a little bit more shorthand now. I kind of ran out of room, so I'll do it over here. So we've got tyrosine, serine, and histidine. And so I'm going to set up that chain first. So I can go, here's my amino group, and then I've got C, C, N, C, C, N, C, C, and then it's a tripeptide, so I'll have my carboxylate on the other end. So that's going to be the same for any tripeptide. Similarly, each carbon and nitrogen that's not the carbonyl is going to have a hydrogen. So I can put in my carbonyls and my hydrogens. So I've got an H, carbonyl, H, H, carbonyl, H, H, carbonyl. And then the only thing left is to add the R groups. So tyrosine, TYR, is a carbon and then a benzene ring, which I am out of room there, but a carbon and a benzene ring, and then an OH on the other side of the benzene ring. What is next? Serine, which is just the CH2 and then OH. And then histidine, I'm not going to have room for this one either, but I'll do it over here. So there's CH2, and then there's this 5-membered ring with 2 nitrogens in it. And double bond here, and a double bond there. So I would draw that a little bigger so you have more room to put in your R groups, but that's how you do that. So as long as you know what the R groups are, you can draw any length of peptide that you want. Obviously, after a while, it's going to get pretty tedious. But that's something to practice. Make sure you understand how those peptide bonds go together. I will give you a table of all of, probably the same one from the book, a table of all the amino acids, so you don't have to memorize those or the abbreviations or anything like that. You have to know how they go together. You have to be able to recognize the different structural bits of proteins and how they come together as well. And what else? And then the forces at play in holding those things together, which are all things we've talked about before. Things like intermolecular forces, hydrogen bonding, dispersion forces. None of that stuff is new. All that's new here is putting it together in this protein structure. All right. Any other questions? Okay, well, we were going to do that activity, but we don't have it, so we'll save that for Monday. So next Monday, do you want to do another quiz on proteins? Yes, no, I mean, we've done a lot of quizzes. But if some people still need some, we can do another one.
It's tough.
But Wednesday,
Wednesday.
next Wednesday. All right, so how about this? I'll get a quiz ready. If you need extra quizzes, come take a quiz. If not, show up at 6-ish, and then we'll go from there on Monday. And on Monday we'll review for the exam, whatever questions you have, whatever you want to go over, and then we'll build these models of proteins so you can see those structures and do that. Then Wednesday will be the exam, and then 1 more week of class, and that's it. It's almost done. Oh, just one thing. I was going through finishing up grades and going through notebooks and things on Blackboard. There's still, I don't know exactly who this applies to, but there's several people where it says that your labs are in progress but haven't been submitted. So if you could check that, because it probably means that you uploaded it already, but you just didn't click the submit button. So if you go in and just submit it, I can't see it until you actually submit it. So I can see that you did something, but I can't actually see the pages until you submit it. So please submit those. Let me know if I'm missing one, or we found a couple that were submitted to the wrong lab, that kind of stuff. Make sure you're getting points for all the stuff that you did. And yeah, 1 more test and the final exam. Let me know if you need anything.